Where to start? Developmental syndromes. I will say at the offset that we have our discussant joining us by Zoom today, so we have some fun technology happening here, but I think it's all going to work out. So just to introduce the topic today, as many of you may know, feeding difficulty and GI symptoms are prevalent in a number of neurodevelopmental syndromes. And this includes both genetic, neurogenetic, neurodevelopmental syndromes, and also conditions like ASD and cerebral palsy that have less clearly defined etiologies. And in addition, food-related behaviors such as food selectivity and overeating and hyperphagia are also observed in a number of neurodevelopmental conditions. And then when we see comorbid obesity or body weight issues, it's often concerning to figure out what are the biological factors and what are the behavioral factors to sort of understand the underlying cause. And all of this kind of speaks to this thing that everyone is talking about now, which is the gut-brain axis and the way that what's happening in the gut affects the brain. Just a couple of snapshots from the media and some minor publications out there about how this may be affecting psychiatric behavior and disorders, autism, and a number of other conditions. So in this symposium, we will be reporting some studies that contribute to further our understanding of potential gut-brain connections in neurodevelopmental conditions by addressing feeding challenges, GI symptoms, and food-related challenging behavior across multiple syndromes. We have an exciting panel. I'm not going to read everyone's title of their talk, but I will go ahead and read the speaker bios, just to introduce the speakers today. So first, Dr. Tues is a postdoctoral fellow in Dr. Margolis's lab at NYU. He earned his PhD in neuroscience from the University of Missouri, where his research focused on the epigenetic mechanisms underlying gene-environment interactions in autism spectrum disorders. He also holds a master's in pharmacology and a bachelor's in pharmacy from University in Seoul, Korea. He has, with extensive research experience in both academia and industry, he's contributed to numerous studies spanning from pharmacological studies to neurodevelopmental research in autism spectrum disorder. And he's going to be sharing a study that has to do with mouse models, which is really interesting. Bacheva Friedman holds an MA in developmental psychology and a BS in social work. She is a clinical research specialist at Boston Children's Hospital, where she helps manage the Angelman syndrome natural history study under the direction of Drs. Wynn Montan and Anjali Zafani and Lynn Bird. She works with investigators and other clinicians to ensure data is accessible and helps make the stories of those affected by Angelman syndrome heard. And she focuses on improving data accessibility, process efficiency, and collaborates on presenting and publishing findings from the Angelman natural history study. Hannah Frawley, my colleague from RTI, holds a dual BA in anthropology and Latin American and Iberian studies from the University of California, Santa Barbara, and is in the process of obtaining a Master of Science in Epidemiology from the University of Lisbon. She has 15 years of international experience, primarily focused on neglected tropical disease control in Africa and child and family outcomes of congenital Zika syndrome in Brazil. And she's particularly interested in dysphagia and related outcomes of children with CEDS, Zika syndrome, as well as caregiver well-being. And she's passionate about using her background in anthropology to ensure research and health interventions are culturally acceptable to diverse populations. Last but not least, myself. I'm Sarah Andrews. I should introduce myself at the start. I'm at RTI International, and I have a master's in teaching from Duke University and a master's in public health with a concentration in nutrition from UNC Chapel Hill. I'm a registered dietitian. And at RTI, most of my work focuses on rare neurogenetic conditions and newborn screening. My primary expertise is in qualitative research methods and incorporating the perspectives and experiences of families affected by those conditions into research and evaluation. And I'm particularly interested in nutrition and feeding issues in neurodevelopmental conditions. I contributed to the presentation that I'm going to give, as well as the report that Hannah's going to present. And we're lucky today to have Dr. Lynn Bird as our discussant. She's joining us virtually. She's on Zoom. She is a dysmorphologist and clinical geneticist and professor of clinical pediatrics at the University of California, San Diego, based at Grady Children's Hospital, San Diego. She's been in practice 30 years and has been involved in Angelman syndrome research for 23 years, and she has personally evaluated more than 150 individuals with Angelman syndrome. Dr. Bird is the PI on several clinical trials aimed at finding treatments for the unmet needs in Angelman syndrome. So we have an exciting presentation. A couple of our talks, the first two, do focus mostly on Angelman syndrome, and then we bring in some other conditions in the second talks. So, testing up.
Good afternoon, everyone. Thanks for coming. My name is Hassan Wu, and I'm a postdoctoral fellow in Dr. Michael's lab at the Pain Research Center at New York University. Today I'm excited to kick off our symposium by introducing an animal model of Angelman syndrome. The title of my talk is Exploring Gastrointestinal Issues in Neurodevelopmental Disorders. Angelman syndrome as a representation. Here's the outline of my talk today. I'll briefly introduce the background of animal of Angelman syndrome with the study goals of my study. And since many of you may be not familiar with animal experiments we use, so I will also explain how we utilize the animal model in the study and the key findings, and then I will wrap up with the key conclusion with future interactions. Okay, the Angelman syndrome is a rare neurodevelopmental disorder caused by a loss of maternal UBE3 gene. Normally, the UBE3 protein is only expressed from the maternal copy. So, when there is a mutation or deletion on this maternal allele, it leads to the disorder. Like many other neurodevelopmental disorders, Angelman syndrome is frequently associated with gastrointestinal symptoms. Among these, the constipation is the most common issue, and gastroesophageal reflux disorder is also frequently reported. So for this study, we used a widely accepted mouse model of Angelman syndrome. These mice have a deletion of the UBE3 gene on the maternal chromosome. Since only the maternal UBE3 gene is active, this results in a loss of UBE3 protein expression, mimicking the genetic cause of Angelman syndrome in humans. So our first aim of this study was to see whether AES mice show gastrointestinal issues such as constipation, reflux, nausea, or abdominal pain. Next, we wanted to figure out which part of the gut of these problems occur, and whether this kind of due to the gut itself or is from brain abnormalities. Ultimately, our goal is to use this mouse model to better understand the pathophysiology of underlying the clinical symptoms to guide the future treatments. So from now, I'll explain the experimental method we used and the key findings. Okay, to understand how gut works in Angelman syndrome mice, we first look at how long it takes for food to pass throughout the whole gastrointestinal tract. So we got non-absorbable red dye and simply waited until the first red food appeared. This tells us how fast their gut is moving, and we found that both male and female took longer to pass the dye, meaning their motility is slower. In male mice, particularly, we also observed that they produced fewer fecal pellets during the observation. This slower motility doesn't directly mean constipation, but it is closely related, and the constipation is one of the most common symptoms in people with Angelman syndrome. Next, we tested how fast the food moved throughout the stomach and small intestine. And this time we got biased the mice with a non-absorbable fluorescent dye, and after 15 minutes, we collected their stomach and small intestine. And we measured how the dye was left in each part. We found that the stomach in AS mice was slower to empty, which means the food remained longer in the stomach. While mice don't vomit like humans, this kind of delayed emptying could be related to symptoms often seen in patients like reflux or feeding difficulty. But when we look at the small intestine motility, we didn't see a clear difference. Then besides the motility test, we also assessed abdominal pain response. We inserted a catheter equipped with the balloon and pressure sensor so that we can detect the abdominal muscle contraction in response to the balloon distension. In male mice, we observed any significant difference between wild type AS group, but in female AS mice, we saw a significant increase in balloon distension response, which means female Angelman syndrome mice exhibited hypersensitivity in visceral pain. Okay, then next, we assessed a intrinsic motility of the gut. So, this apparatus, we placed isolated gut in the organ bag, and this setup allows the gut to keep contracting on its own. Without any influence from the central nervous system, is contracting solidly from the enteric nervous system. By recording these contractions, we could evaluate gut motility only driven by the enteric nervous system. We recorded these videos and converted it into contractions heat map. And then we first took the large intestine part. So these are the representative images of large intestine motility heat maps. And from our results, the frequency and velocity and the length of the large intestine contraction showed no significant changes between the wild type and Angelman syndrome mice. This indicates that the large intestine motility driven by the enteric nervous system is preserved in Angelman syndrome mice. We also measure the intrinsic motility of small intestine, particularly the most proximal part of the small intestine. You can see since the contraction pattern of small intestine looks somewhat different from the large intestine contraction. So we use different parameters to validate their contraction pattern. So in male AS mice, we observe a significant reduction in contraction amplitude, which means the small intestine of Angelman syndrome mice, the contract weaker than wild-type mice. Other parameters like duration or frequency didn't change a lot, and there's no difference in female adolescent mice and compared to the wild-type mice. And to better understand the mechanism behind infertility issues, we also analyzed the number of neurons in the gut. The enteric nervous system contains two major neuronal layers, the myocardial plexus and submucosal plexus. In this study, we focused on the myocardial plexus, which regulates the motility. We use a specific antibodies to stain. Sorry, it's not very clear. We use stain antibodies to stain the neuronal cell bodies in red and UV3 protein in green. As you can see, not clearly, but yeah, the UV3 protein expression in the enteric nervous system in Angelman syndrome mice is remarkably reduced. So we found a significant reduction in neuron number in duodenum, the most proximal part of the small intestine, and stomach of male Angelman syndrome mice. This is the region-specific and sex-specific neural loss may help explain the kind of impaired gut upper GI motility. So lastly, I'd like to just briefly mention that one of our collaborative groups conducted from UC Davis, Dr. Melanie Garwood's lab. They found out there's a decrease in the secretion of the gut, and also they found out the microbiome alteration in Angelman syndrome mouse, various animal model. So they also found that there is a short chain fatty acid reduction, which may contribute to the constipation. To summarize, we found that ACE mice have slower gut transit and late stomach emptying. This was linked to the neural loss in upper gut, specifically the duodenum in both sexes and male in young stomach. So we also saw a difference in the gut motility and sex-specific difference in visceral pain motility, and which requires a further study. To wrap up, since we have identified neural loss in upper GI tract, the next goal is to define which subtypes of enteric nervous system are most affected. And next, we plan to perform region-specific UV3 expression mapping, and we will also investigate the potential genetic pathways interacting with UV3 gene. And we are doing collaborative studies with Angelman syndrome natural research investigators. They're Dr. Wenan Basheva and Dr. Lieber and other comments here, thanks to the whole group. So, yeah, I'd like to express my gratitude to all my lab members, Melanie and all collaborative groups. Thanks for coming here and listening. Thank you all for listening.
Are there any, we're going to go take one or two questions after each talk. If anybody has a question and there's a mic in the room.
I'll just ask one quick question.
Sit down.
I'm also wondering if maybe the microphone over there might not turn on, because of the static. I'll just talk loudly. There's not many of us here.
It's on.
Yeah.
Oh, I'm sorry. I was just wondering if gender differences that you saw, the sex differences that you saw,
Yeah.
are those also seen in other disorders like autism, or this is not my field, so I don't know.
Yeah. I haven't looked at any developmental disorder, GI disorder in animal model, but yeah, I think from the literature study, yeah, there's been sex differences. Not saying like this animal syndrome, there's a kind of pain difference, sex difference, but yeah, not sure that there could be a motility difference in animal syndrome, the other developmental disorders.
Okay, all right.
you considering supplementing with short chain fatty acids? Like, so more phenylbutyrate is in.
Yeah, actually, in UCLA, Melanie is doing that, the butyrate, supplementation or so. Yeah, this is one of the our collaborative studies.
Because it could have implications for many other developmental disorders and other symptoms. So
Thank you.
Hello. And did such a good job introducing me. So I'll be sharing some data from the Angelman syndrome natural history study today. So as the name suggests, the Angelman syndrome natural history study is an observational study. That means there's no therapeutic, no experimental drug. It's longitudinal, so we meet with the same participant on multiple occasions, and it's long term. So we try to follow them for as long as possible and as long as they're able to stay in the study. During the study visits, we do full battery of assessments. We do Bailey scales of infant development, we do environment adaptive behavior scales. We do a complete physical and neurological exam, and then we also collect some retrospective data, so medical records, genetic testing results, and then a comprehensive initial medical interview. So the study has been ongoing since 2004, and it's evolved throughout the years. It was originally funded through NIH and then supported for a period by the FDA, and we're currently funded through ABOM, which is a consortium of patient advocacy groups and pharmaceutical companies and other stakeholders. You can see that our enrollment patterns have shifted over the years. The study saw the largest wave of new enrollment during the earliest phase of the study, and we're currently prioritizing follow-ups with these participants. We currently have 9 sites, which you can see on the map across the US and Canada, and the little airplanes represent all site destinations, so sometimes the main study team will travel to other areas that would not otherwise be represented in the study. We're actually headed to Texas in a couple weeks. So a little bit, you heard from Tessa about what we know about GI issues in Angelman syndrome. We know that constipation and GERD are common, but poorly understood. We also know that seizures can be quite common and that both conditions significantly impact quality of life. We also know that GI symptoms can persist despite multiple treatments, and similarly, seizures can require multiple treatments, including medications, diet modifications, in some cases surgery. There's limited understanding of how these symptoms and conditions interact, which makes treatment very difficult. And so we're so grateful for this collaboration with Payson and the Margolis lab, showing how the loss of PDE3 disrupts gut motility in Angelman syndrome mice. And we're currently working to correlate their findings with our data to advance care for Angelman syndrome. So for this presentation, we're looking at data from a subset of Angelman syndrome natural history study participants who completed the most recent version of our GI and seizure questionnaire. And this figure just shows the distribution of male and female participants for the different molecular diagnoses. So on the x-axis, you can see the different molecular subtypes, starting with deletion, which is the most common and also the most severe, followed by mutation, and then the more rare forms of PD and protein defect. And then on the y-axis, you can just see the percentage of males and females for each subtype, and it's fairly split across the sample. Moving on to the age of the participants at their most recent visit. So this just shows the median age along with the 25th and 75th percentile. And so you can see that overall, the median age for participants is about 10 years, 25th is about 5 years, and the 75th about 20 years. Our youngest participant was about 1 year old, and our oldest participant about 52 years old at their most recent visit, and similar distributions across the subtypes. So one of the more prevalent GI-related symptoms with early onset is feeding difficulties during infancy, and this figure just shows the percentage of participants from each molecular subtype who reported feeding difficulties, which is represented by the color yellow. And you can see these difficulties are common across all the subtypes, with individuals with deletion being particularly impacted. And another early GI symptom we often hear about is failure to gain weight. although this tends to be reported to a slightly lesser degree. And this next figure again shows the percentage of participants across the different subtypes who reported difficulty with weight gain and immunity. So shifting gears to the more recent visit, this next figure shows the percentage of participants from each subtype who experienced gastroesophageal reflux disease or GERD, which is a condition where the stomach acid flows back into the esophagus, commonly known as heartburn. For individuals with the deletion, close to 50% of participants were experiencing GERD, and it's also quite common in other subtypes, although not as prevalent. So medications commonly prescribed for GERD in this sample, include proton pump inhibitors, which block the viral step of acid production. These are usually prescribed for longer lasting relief. I'm sorry. Thanks for bearing with that. We also see H2 blockers prescribed fairly often. These also reduce stomach acid by blocking histamine receptors. They're faster acting but less potent. And then antacids, which are usually prescribed to neutralize existing stomach acid and are used for more quick short-term relief. So the next symptom we looked at was constipation. And to assess severity, we used a composite score based on the bowel movement frequency over the 4 weeks leading up to the visit. So fewer bowel movements would indicate someone is more constipated. We also looked at stool consistency, so harder stools would be scored higher. The number of constipation medications actively being used, and then based on total scores, participants were characterized as having no mild or moderate or severe constipation using percentile cutoffs. So here you can see the results, and on the x-axis, again, we have our molecular subtypes, on the y-axis, percentage of individuals in each constipation and severity category. So starting with the deletion group on the left, you can see that the vast majority reported some degree of constipation, only 18.5% reporting no constipation, and about 14.8% experiencing severe constipation. And we see a similar distribution in the mutation and the UPD and imprinting defect group. Most individuals experience at least some constipation, mostly mild to moderate. Some of the treatments commonly used for constipation in the sample are stimulant laxatives. These trigger intestinal muscle contractions to promote bowel movements. We also see the use of osmotic laxatives, which draw water into the intestine to soften the stool. Marilax is the commonly known one. And then occasionally we see the use of enemas, which introduce liquid into the rectum to clear the lower bowel. And then a lot of our participants are also using dietary supplements. So this next figure shows the number of medications taken to manage constipation, and it's broken down by subtype. The green bar represents participants taking no medications. So across the full sample, many participants are taking between as many as 1 to 4 medications just to manage their constipation. The UPD and imprinting defect group have the highest percentage of participants not using any medications, but even within that subtype, it's very common to be taking some medication to manage these symptoms. So after taking a look at GI issues, we shifted our focus to the experience of seizures during the same 4-week window leading up to the visit. So here we asked caregivers to report on the number of seizures their child experienced. They're classified as mild to severe. What makes a seizure severe is that these seizures typically involve motor activity, jerking, stiffening. They could have generalized onset, loss of consciousness, and therefore a higher risk for injury. So, looking at the percentage of participants who experienced mild seizures within this 4-week window, we see that most participants reported no seizures, which is the blue bar. And of course, this could just be by the virtue of the fact that if they're coming to a study visit, then they might not be coming if they're experiencing seizures. However, we do observe that some participants experienced multiple seizures, with over 21 seizures reported by several participants. And similarly, for the severe seizures, we see that almost none of our participants, thankfully, reported experiencing seizures in the 4 weeks leading up to their visit. And again, this could just indicate the sampling bias, and also that the seizures are well controlled right when before they show up. So what is striking is that despite the reports of low seizure frequency, when we look at the number of active medications being used by this cohort, we see that starting with deletion on the left, only about 20% are not taking any medication. The majority of participants are taking anywhere between 1 and 5 medications to manage these seizures, and this could suggest that the medications could be masking or really treating their symptoms, despite the low report of seizure frequency. Some of the commonly prescribed medications used in the sample are just classic AEDs, benzodiazepines, CBD-based treatments, often used as an adjunct therapy, and then dietary modifications like a ketogenic or low glycemic index diet. So a bit about what we learned and what we still want to know. So we know that in Angelman syndrome, GI symptoms can begin early, that GERD and constipation are common for all subtypes. Seizures are occurring less problematic, but that could also just be due to the fact that the symptoms are mostly managed. And we also see that managing all these symptoms is complex. Participants could be on multiple medications at once. So some limitations, we are only looking at one snapshot in time for these participants. The treatment can be masking the severity, and we don't fully understand the interaction between some of these medications, which a known side effect is actually constipation. And then we're also just dealing with caregiver-reported data. Seizures can be very hard to identify and classify. And so what we want to do next is expand this data set to look more longitudinally for these participants and capture more time points and look at how different medication types and classes affect the symptoms. We're also going to look into some alternative seizure metrics like the duration of the seizures, intensity, age of onset, and then a new project. We're going to have caregivers submit videos of seizures as they occur at home, and the clinician can help classify and diagnose them. So thanks to everyone who contributed to this work. Special thanks to Dr. Bird, who actually started the Angelman syndrome natural history study almost 20 years ago. And we're lucky to be hearing from her today. And Dr. Tan and Savani, who helped maintain the study. These are all the site principal and guest investigators, and thanks to our funders, and of course, the Marco List Group for their collaboration. And most of all, to the families who over all these years have been contributing and really educating us on what it's like having Angelman syndrome and how to better manage their care. Thank you.
Any questions for Dr. Jeff? Okay. On feeding practices and weight status of children with congenital Zika syndrome in Brazil. So just a very quick background on congenital Zika syndrome. You might remember the Zika outbreak in 2015 and 2016 that affected all of the Americas, but mostly Central and South America. During this time, approximately 3,000 babies in Brazil were born with microcephaly that was later found to be linked to the Zika virus and led to congenital Zika syndrome, the discovery of congenital Zika syndrome. The main or most commonly known characteristic of congenital Zika syndrome is severe microcephaly at birth, but also these children have other brain abnormalities, including ossifications, a very specific ocular finding, and congenital contractures and hypertonia and spasticity. Research in the first couple years of their lives show that these children have lifelong functional impairments and increased health risks, profound intellectual disability, and feeding difficulties. And in those first 2 years, this was specifically around difficulty with breastfeeding
and more severe nutritional status and dysphagia, or the difficulty with swallowing. However, little was reported on these outcomes in children as they aged beyond 2 years of age. So today's data comes from Project Seminar, which was a longitudinal study of children with confirmed or presumed congenital Zika syndrome funded by the Fertility Center at the NIH. The study is a collaboration between RTI International in the US, led by Dr. Ann Wheeler and Dr. Don Bailey, and the Altino Ventura Foundation in Recife, Brazil, which was the epicenter of the congenital Zika syndrome outbreak in Brazil, led by Dr. Camila Ventura. The project data collection period lasted from 2018 to 2022. During that time, families came in 13 times, 13 site visits. Those were divided and alternated between the child-focused aims, which looked at the developmental and functional outcomes of children across those 5 years, and more family-focused outcomes, including family adaptations and caregiver well-being. So the data today comes from the 6 child visits, which you can see at the bottom. At the first child visit, the average age of children was 32 months, and at the last child visit, September of 2022, the average age was 84 months, which is under 7 years. However, the mean cognitive age of these kids, as measured by the Bailey scale of development, was 2.7 months at first visit and 3.3 months at the final visit. So the data for the feeding practices came from a questionnaire that was developed by the research team at RTI and FOG, and this questionnaire captured information that was reported by the parents on a wide variety of feeding practices and outcomes, like the use of an alternative feeding method, both the type of method and whether the child used it part of the time or exclusively for all of their food intake, symptoms of dysphagia, both while consuming liquids and solids, the consistency of the food, are they able to consume solids, do they need their liquids to be thickened, and other feeding behaviors like the duration of their meals, they have any specific food preferences. We also measured anthropometric data at each of these visits, and specifically here I'll present weight. As you can see in the photo, most of the kids are in wheelchairs. I think over 90% of the kids in our study are in wheelchairs. And the foundation at this moment doesn't have a wheelchair scale. So in order to take the weight of children, we measured the weight of the caregiver holding the child and then subtracted the caregiver's weight to get the child's weight. Our population has pretty low literacy levels, which is common in Brazil, and so all of the questionnaires were administered directly by trained therapists at the foundation. They read the questions and the response options and then entered the responses directly into RedCap. So I forgot to mention that the data here is from 156 children of 208 that enrolled in the study, and we limited it to 156 children that had complete data at the first 4 assessments. And of those 156, 54% were female, 17% were born prematurely, 27% had low birth rate, and 84% had microcephaly at birth. 65% had Zika virus exposure confirmed by serology. What this means is that 65% within the first 3 months of their lives were able to have access to a test to confirm that they were exposed to Zika virus in utero. That doesn't mean that the other 35% had a negative test, it just means that they didn't have access to the test, because at the time there wasn't widespread testing. And so those 35%, we can't say have confirmed congenital Zika syndrome, but we classify them as having presumed congenital Zika syndrome based on having the clinical features that I described earlier, as well as having laboratory evidence that rules out any other infectious cause, such as toxoplasmosis or CMV. We tested for a difference in outcomes between the 2 groups just to see if there was any, and we did not find any significant differences. And so we were presenting all the data for all children with both confirmed and presumed together. So what were the main outcomes? Unfortunately, we saw that the feeding abilities of these children are declining over time. Starting at the top, you can see that almost 3/4 of children at the start of the study had a symptom of dysphagia while consuming liquids, and that increased to 85%. Half of the kids could not control their saliva because of dysphagia, and that increased to 71% of the kids by 10.5. At the start of the study, 20% of kids were using an alternative feeding method, and that increased to 1/3, 33% by 10.5. And And then consumption of solid foods, or the ability to consume solid foods, decreased from 43% of the sample being able to consume solid foods to only 31%. All of these findings were found to be statistically significant across the 5 years. Time point 6 is not included in that analysis because, as that was the final study assessment before the conclusion of our funding, we only reached half of the sample, and so the frequencies are shown here, but they are not included in the analysis. But you can see from the frequency that, with the exception of the control of saliva, the trend is continuing. So we can anticipate that with such profound difficulties in feeding, we might see an impact on the child's weight. So we calculated weight for age Z-scores using WHO's Anthro Plus program, where underweight was defined as being greater than 2 standard deviations below the mean for their age, and severely underweight was found as being negative or more than 3 standard deviations below. We found that this was pretty stable across time. So while we saw a significant decrease in feeding abilities across time, we did not see a significant decrease or increase in the weight status of children. It remained relatively stable. So across all time points, over 40% of kids, just over 40% of kids were considered to be underweight, and 25 to 30% were considered to be severely underweight. We then looked at we used logistic regression models to test the association between being underweight and the exclusive use of an alternative feeding method, and found that there was a significant decrease, and the odds of being underweight were 70% lower in kids that have an alternative feeding method. So I was going to give a little story about why this is so important, not just for us as researchers, but for the families that are in our study. This is a girl in our study. You can see on the far left. She's very, very tiny. This was when she was about 5 years old. Her weight was just under 12.5 kilos, which puts her in the severely underweight category based on her weight for age. A few months after that photo was taken, her parents decided to give her a feeding tube or have a feeding tube placed. And you can see it in the middle picture, which is just a few months after having that feeding tube, her weight had already almost doubled, and her weight for age Z-score came to 0.1, so right, pretty much on par with the mean for her age. About a year later, after the placement of the G-tube, we were still seeing, and are still seeing, a pretty steady growth and healthy growth in her. This is important because a lot of families in our study really struggle with the decision of when and if even they should place their child on an alternative feeding method. There's a lot of fear. They fear that it could be painful for their child. They fear about the risks associated with the surgery and anesthesia. They fear that they don't have the adequacy to take care of a child if they have a feeding tube. And they also just worry a lot about the quality of life for their children. So this mom is a really, really local mom in our group, and she really took it upon her to share her experience with the other families. And I have a quote from her here explaining how having a feeding tube really changed the life of her daughter. She now is a different child. She's alert. She goes to the therapies and seems to be thriving in her therapies. And other parents hearing that has been really instrumental, in my opinion, from what I see, in helping to increase the use of feeding tubes in this population. So our main conclusions from this study and this research is that children with congenital placenta syndrome do experience worsening difficulties between 2 and 7 years of age. Most of them are declining. We do see some kids show some improvement. We also know that dysphagia increases the risk of undernutrition and aspiration, which I didn't talk about much here, but obviously if they're having trouble swallowing, they're also at risk for aspiration pneumonia, and that is one of the leading causes of hospitalizations and mortality in this group. But we also know now from this research that the alternative feeding is a good intervention to help improve those nutritional outcomes. And so it's really essential to work with families to help them make those decisions on what's going to be best for their child. So next steps, we are really excited that just last year we had a new award provided for 5 more years to follow these kids. We're going to continue our collaboration with the Altino Ventura Foundation in Brazil. We're also bringing in the University and the University of North Carolina to help some specific research aims in different areas not related to feeding. In terms of the feeding and nutrition, what we hope to see going forward with this new study is that we will be working with local clinicians to do more in-depth assessments, specifically with nutritionists, gastroenterologists, and endocrinologists. They will be looking at using 24-hour recalls to measure the actual nutritional content of the children and their diets, using DEXA to assess the total body composition of the kids, as well as using labs to look for an endocrine disruption or metabolic alterations that might be contributing to changes in food status. And then I was just going to end on a small anecdote about the name of the project, Project 70R, which Portuguese means to sow or to plant. But it also is an acronym for the study where each letter represents a specific characteristic that the project hopes to sow or to plant in the families and in the community. And those are solidarity, hope, motivation, emotion, love, and resilience. And I personally think that we've been successful. We had 92% retention at the end of the first phase of the study, so we are really looking forward to continuing to sow these great characteristics in the community. Thank you.
I'm happy to adopt you, Mike. That was a fantastic presentation. I'm so interested in the use of the GQ at such a later age. I work in cardiology where literally as soon as they're born, before they go home, all our kids have G-tubes if they cannot feed. And so I'm just curious, are there cultural considerations or cultural differences in the acceptance of G-tube?
So just anecdotally, I would say yes. I think food is a huge part of responding culture, and so I think parents, that is a big thing in the food, and the family eating and continuing together, and so I think that's a big part of it. It's just not having your child be able to participate in that, and getting meals in that way. It's not something that we've actually looked at and studied, but we are looking into some more qualitative research, and this is a great thing that we could explore, and I think especially knowing what we've heard over the last couple of years about the resistance in families and hearing more about how those families are making the decision and changing their mind, I think it's been really useful for the community going forward. I think just one other note since you mentioned with the age, we do see some kids that kind of fluctuate in their use, and so they might have used it when they were really little and then stopped and then went back. And we also see that the change from a more temporary solution like a nasogastric tube to the actual placement of the PEG tube does increase as the kids age. Any more questions? Thank you.
So I'm going to be talking about specificity of early childhood hyperphagia profiles. And I'd like to thank my collaborators here from the Caliber Lab at Purdue University and my colleagues from RTI that are here, and especially wanted to give a little shout out to Sarah Potter who helped bring these panels together. So hyperphagia, as you can see here in the image that's projected, is an intense, persistent sensation of hunger accompanied by preoccupations, an extreme desire to consume food and drive to consume food, and it can cause other food-related problematic behaviors, acting out, etc. Generally, it's a lack of satiety. And I chose this. This was shared by the Foundation for Prader-Willi Research last May during Prader-Willi Syndrome Awareness Month. And I picked it on purpose because everybody thinks of Prader-Willi when they think of hyperphagia. However, in the past several years, there have been increasing reports of hyperphagia in other neurogenetic conditions, including Angelman and Fragile X. We know that these conditions are distinct for sure, but there are common phenotypic features such as intellectual and developmental delay, autism symptomology, sensory sensitivity, challenging behaviors occurring in varying severities and to different degrees. And so, we were interested in learning about where hyperphagia fits into sort of the spectrum of things that can occur in multiple neurogenetic conditions and how it relates to other clinical and developmental features in these conditions. So the questions we looked to answer were, how do hyperphagic symptoms differ in early childhood across different neurogenetic condition groups compared to controls, and then how do hyperphagic symptoms relate to other clinical and developmental features? Thanks to Dr. Bridget Caliber, I don't think she's in here, but she's here. We were able to use data from the Purdue Urban Phenotype Study, or PEPS, which is a longitudinal caregiver-reported survey study of children with neurogenetic conditions. In this particular sub-study, we focused on Angelman syndrome, Prader-Willi, and Williams syndrome, and mostly just because of numbers of sample size, being able to draw comparisons, as well as a group of low-risk controls, or kids who don't have a known condition. And we refer to them as LRs throughout these slides. So caregivers complete measures at different time points on a schedule, and starting at age 4, they complete the hyperphagia questionnaire. It is an 11-item Likert scale from 1 to 5 that addresses symptoms of hyperphagia, and a score of 5 ends up being like the more frequent or the more severe end of whatever the question is asking. There's a raw total score that's calculated, as well as 3 domain level scores. So I'm going to just briefly share what the items are in each of the domains to give you a sense of what the parents were asked about their kids. So there's the behavior domain, that's 5 items that address actions that one takes to obtain food. There are 4 items in the drive domain that address the degree to which one is focused on food. So you can see that it has to do with being persistent, being able to be redirected. And then 2 items that address severity, the severity domain, the degree to which thoughts and behaviors associated with food interfere with functioning and daily life. In the PEP study, and in this particular sub-study, they also assessed other symptomology. So we looked at measures that assessed autism symptomology, sensory-related behaviors, challenging behavior, and adaptive functioning. I'm sure that many of you are familiar with these measures, but kind of wanting to assess the relationship between these and the hyperphagia questionnaire as well. So this is our cohort. I'm just going to draw your attention to the sample sizes being close enough to draw statistical comparisons, and really the only statistical difference across the groups in any of these demographic type characteristics was in BMI. The BMI of kids with Prader-Willi was significantly higher than low-risk controls, and the BMI of kids with Williams syndrome was significantly lower than both low-risk controls and kids with Prader-Willi. So first, we compared the total HQ hyperphagia questionnaire scores and the domain level. We compared each neurogenetic condition group to the low-risk controls, and it's a little bit hard to see, I think, on this graph where we see significant differences, so I put some red in here. But you can see that, interestingly, all 3 of the neurogenetic condition groups were significantly elevated compared to low-risk controls in total hyperphagia scores, as well as behavior domain. Angelman and Williams syndrome were elevated compared to low-risk controls for drive, and only Prader-Willi was elevated in the severity domain. And here, we're kind of getting a little bit granular. Bear with me, I'll walk you through the presentation here, but we can still see the domain level, the blue, sort of those top bars, and then these are the items within each domain, and the symbols indicate significant difference between each neurogenetic condition and the low-risk controls at the item level. So some interesting things to call out here. Both Prader-Willi and Angelman syndrome showed elevations in 2 of the same behavior domain items, foraging through the trash for food and being clever and fast at obtaining food, as well as a drive item, distress when told to stop, related to awkward behavior. Only Prader-Willi was elevated in a few items, or a couple, trying to steal food, as well as both disparity items. And interestingly, Williams syndrome, while elevated at the domain level, was not elevated at the item level after doing correction for multiple comparisons, the on-run correction. And then looking at the other clinical and developmental measures, what we found is that the mean scores on these measures were significantly greater than low-risk controls, indicating more severe symptomology for all the clinical measures, and significantly lower than low-risk controls in the Vineland Adaptive Behavior Scale and some some scales indicating lower adaptive functioning, which is what we expected. But we then wanted to look at, sorry, I'm trying to figure out how to scroll down here. Yeah. Can't do it. Okay. We then wanted to look at the relationship between some of these other clinical symptoms and hyperphagia. So we ran correlations between the score measures and came up with a model. And when we examined, in the model was the CBCL externalizing SCQ and the Vineland adaptive behavior composite score, as well as we had variables for each of the neurogenetic syndrome diagnoses. So when we examined these predictors in a stepwise regression, the model of best fit showed that the CBCL externalizing behavior and Prader-Willi syndrome diagnosis were the biggest predictors of hyperphagic symptoms or total HQ scores. And this figure is showing what we saw when we controlled for the CBCL externalizing behavior. So on the x-axis we have, my goodness, no. We have the externalizing score, and on the y-axis we have the HQ total score. And when we controlled for externalizing behavior, only a Prader-Willi diagnosis was associated with significantly higher HQ scores. So interesting. Overall, we found that the presence of hyperphagic symptoms is not unique to Prader-Willi syndrome, but it varies across the groups that we looked at. We see different items, different behaviors being endorsed, and overall it seems that hyperphagia still appears to be more severe in Prader-Willi, indicated both by it being the only group that was elevated in the severity domain, and also the fact that when we controlled for other clinical features, the hyperphagia symptoms were only significantly elevated in Prader-Willi syndrome. In terms of next steps, it would be great to update these analyses with new data that's coming from the PEP study, and including additional conditions that we didn't have large enough sample sizes for. Additionally, I am extremely eager to do qualitative research because we would love to understand some of the findings that we were surprised by. One thing to think about is that individuals with Williams syndrome can be very communicative, and we wonder whether the drive endorsement is indicative of just an overall perseveration on the topic at hand versus having an underlying etiology of satiety and hyperphagia. Also, we saw Angelman syndrome being elevated in an item that had the word talking in it, and most of that population is nonverbal. So we're eager to understand what parents are seeing when they're endorsing some of these symptoms. We also found, I didn't get into this much, but one of the items about stealing food was not answered by, I think, 2 low-risk control parents, 2 Angelman and 2 Williams syndrome. And so we wonder a little bit, like, that could be anything. They could have just skipped it, or it could have been that they didn't like the word steal, or it could have been that they felt not applicable to their child. So I'm really interested, given that we seem to be seeing reports of these symptoms and other conditions, making sure that we have the right measures to assess them. I should have mentioned earlier, but the hyperphagia questionnaire was developed for and validated on Prader-Willi. So it's been used on other conditions, not just in this study, but I'm eager to make sure that we sort of understand whether we're asking all the right questions and we assess this further. Big thanks to our funders and all the caregivers who participated, and that's all from me. Questions? Okay. I'm going to try to, one second. Lynn, if you want to be seen, I think that I can make you visible to them. Just give me 1 second.
Well, can you hear me?
We can hear you. So why don't you?
More important probably, that you should be able to hear me.
Yes. Go ahead.
Hi, it's very kind of Sarah to have invited me to offer some comments. I don't have much in the way of lengthy comments to make, but I hope that maybe some of what I say might have might be a springboard for additional questions. So Dr. Wu presented some of his work done in the Margolis lab, and I happen to know that there's lots more that he left out. So he's done a tremendous amount of work to provide some insight into the GI symptoms that we see in people with Angelman syndrome through his mouse model. And as all good research does, it brings up as many questions as it answers. So the differences that he demonstrated between male and female mice are prompting us to look at our natural history data for sex differences in GI complaints. It hasn't been apparent to me on just casual observation that I hear more about constipation in one sex versus the other, but I think it's something that we need to look at in people living with Angelman syndrome. His work showing slower gut transit in Angelman mice also suggests that maybe drugs which promote motility might be a more effective path to treating constipation than osmotic agents, which I think are currently more widely used in this population. Bathsheba's data show that we have a long way to go to be doing a good job treating constipation, since the vast majority of individuals have at least moderate constipation, but there's a large percentage of individuals that are taking no medications or report taking no medications. So I think we need to do a better job of making this a priority for symptomatic treatment. I really found Hannah's data on congenital Zika syndrome surprising, because if you'd asked me to predict whether symptoms would get worse or better over time, I would have guessed, of course, they were going to get better, because I think of this as being an insult at a single point in time during gestation. And then I would have expected that you'd go from there and see basically continuous improvement. So I was really rather surprised at the worsening of feeding, in the early childhood years. And I'd be interested to know if this worsening of feeding is basically all due to oral motor skills getting worse, or what component is driving this. And if it is oral motor skills deteriorating, is there deterioration in any other domain of development during that time? Or are these children, other than feeding, basically pretty stable in terms of their level of functioning? So maybe Hannah will address that for us. And then lastly, Sarah's data on hyperphagia I find really interesting. There's certainly, I would love to know if the subtype of the Angelman children that were enrolled was gathered, and whether or not you had enough numbers, I suspect probably not, to be able to divide the Angelman children by subtype. My casual observation, just from having talked to families, is that you mostly see the hyperphagia in the children with uniparental disomy or printing defect classes of Angelman syndrome, and that it's not a big component of children who have deletion or UBE3A mutations. But that casual observation again needs to be borne out by looking at our natural history data to see if we really can tease out. We do have very few questions that deal with apparent increased appetite and hyperphagia in the natural history study, but we probably, it's a much cruder way of looking at it than something more formal like the HQCT. But we might have enough data to get a little bit of insight into whether or not it's particular genotypes that are driving that. And then the last comment I'll make is that I worry about using the HQCT in Angelman syndrome just because I think the motor difficulties and the communication difficulties that these children have may prevent them from fully acting upon any hyperphagia or drive for food that they might have, simply because they can't communicate their needs well enough. And I hope when the instruments were, or when the questionnaires were administered, that it was made clear that it's not just verbal communication we're talking about, but any mode of communication, requesting food or repetitively asking for food. So anyway, those are my comments. I would love to hear from anybody who has any insight into some of the kind of random questions I threw out. And thanks once again for the opportunity.
Lynn, can you hear me when I talk into this microphone?
I can.
Okay. Oh, I don't know. That might be a bad idea.
Is that turn off? Can you turn off that one if it's not turned off?
This one?
Yeah, if it's not turned off, maybe.
Try something. Why don't we just stand up and do it here?
Do you want to use this one?
Go this way and then we can see us. Do it first. Okay, I'll answer questions. On mine first. First of all, I'm just thrilled that you could join us and then it worked out, because I feel like it's the perfect complement to what we talked about. So you asked a couple of questions about mine. The PEP study does have the genetic subtype, but as Lynn suggested, the sample, we didn't have enough, or we didn't have it on everyone, do you remember? Yeah, it was missing for some, and overall it wasn't enough that we had, or that we had large enough numbers to compare across the two subtypes, which is another reason why I would love to do a larger study. And I just wanted to mention, so Lynn mentioned the HQCT, and I talked about the HQ. They are slightly different. One that the HQCT is for clinical trials. It was sort of like the next phase, and they removed 2 items that were determined to be not as observable for use in clinical trials, but the PEP study uses the original HQ, so that's what we reported on. I definitely agree that it might not be the best fit for Angelman for the same reasons that Lynn brought up. So trying to get some funding to do some qualitative work and measure refinement for Angelman in particular. Hoping that happens. And I think that there also has been movement to analyze some of the natural history study data on the feeding behaviors that they've been collecting. So I'd like to see kind of a convergence of those things moving towards figuring out exactly what's going on in Angelman. Is there worsening, deterioration? Is there deterioration in other domains?
Yeah, I can definitely look at it. Thank you for those questions. I think most of them were just kind of generally around the deterioration, and that being surprising us, and of course we hope that they would be improving. So I think to the first point about the exposure being one point in time and hoping that maybe they would improve after that exposure has gone away. That is something that we are trying to look into under the new study, as we are working to assess the evolution of the structural features that were characteristic of the children at birth, and so we're specifically looking at MRIs, and we have a neurologist that we work with in Brazil that has done a little bit of research and has seen that meaning there's fluctuation across all of the domains. Overall, a majority seem to be declining, but there is a number that we see improvements and there's a number that we see stability. And that is something that we also are continuing to look at across each of those very specific study aims. That is another aim of our new study, is to look at what are the predictors of outcomes. And so we'll continue to look and try to understand more what is contributing to these better outcomes. One of the things that we, or just to note that the one slide I showed was the age of the children at the assessments, just to go back to that, and the fact that they were about 2.5 when they enrolled, that had a cognitive age of around 2.7 months, and that was pretty consistent across the various domains of the Bailey, so including communication and motor. They were around that 2 to 3 month age equivalent. And for the cognitive age, that increased to only 3.3 months at the 6th assessment when they were about 7 years old. So there was a very small improvement, but I can't say right now whether or not that was a significant change or not. But also noting that within our group of 208 kids, 90% of them or so have that more severe presentation. We do believe it's the largest cohort of kids with that severe presentation of congenital Zika syndrome, but we do have a small group of about 5 kids that are less severely impaired. And I was trying to look up their cognitive ages. I believe they were more in the 19 months to 36 month range of looking at Yeah, so they definitely are doing a little bit better, and we are seeing a little bit more consistent improvement in those kids. And then one last note about fluctuations and possible reasons for decline that we want to perhaps explore more is access to services, and really looking at what kinds of services are these kids getting, and is that making a difference in how much they're able to improve. And also noting that during the middle of our study, the COVID pandemic hit, so all of these kids lost access to their services for about, we can vary some amount of service, but for a pretty good chunk of time. I think I touched on your points, but let me know if there is anything else that you have questions.
No, that's great. One other thing that occurred to me, Sarah, is that there's no evidence, or I shouldn't say is there any evidence of any ongoing damage? In other words, the infection happened, the infection's over, and you're now just left with the aftermath? Or is there any ongoing, is the immune system still doing something negative?
That's a great question. I think we don't really know, which is why we wanted to get opportunity with this new funding to get those repeat exams. So looking at all of those 5 clinical features, so the MRIs to look at the neurological outcomes and have those stay consistent, or those deteriorating or improving. Ocular exams to look at the ocular findings, and then the kids will also be seen an orthopedist to measure their orthopedic fractures, scoliosis, and hip luxation that they have as well. So I don't think we know the answer, and it is something that we'll be able to answer in the next couple of years.
Fantastic.
Questions and comments for anyone?
Oh, yes. I have a question. So I was curious, as that Shiva mentioned, the age within children in the studies often are on multiple medications across several domains, and I was curious if you thought that being on those medications, or a number of them, would have any impact on the hyperphagia and presentation in children with Angelman syndrome.
Oh, that's such an interesting question. Did you hear the question, Lynn?
Can you repeat it? It was a little faint.
Yeah, she was noting that the kids with Angelman, or the individuals with Angelman rather, are just talked on multiple medications, and I'm wondering if thought that the medications could be affecting the hyperphagia results, or could they influence whether or not people are having hyperphagia, symptomatic.
Yeah.
So yeah.
Certainly there are some psychotropic medications, like I think Abilify is fairly notorious for people putting weight on. I don't think necessarily of the seizure medications as being problematic in that area, but I have not just been casually aware of any sudden change, let's say, in somebody's food drive when they started new medication. So I've never associated it with medications. And the patients I'm particularly thinking of that are seen at my site in the study aren't on medications for seizures because that hasn't been part of their presentation so far. It's worth looking at because again, this population is also on stuff for sleep because sleep is a major problem. I think it would be great when we have a big enough number, which I think we were getting to be able to look at those multiple different factors and see what correlates with the hyperphagia.
Yeah, I think that's really important, and I'm aware of a case study, and I think both Prader-Willi and Angelman, they've used GLP-1s and shown improvements in behaviors, and so that's kind of the opposite direction maybe of what you're thinking of, but I'm interested to see study that more too, what's behind that.
I'm aware of a very small phase 2 study of a GLP-1 in Prader-Willi that was in one of the first generation GLP-1 agonists, and it did not show apparent benefit. But I think it's certainly worth looking into the newer generation GLP-1 agonists. And I know there's anecdotal reports of improvement in an individual with Angelman syndrome.
Yes. For our next round of research. Okay. Well, thank you all so much. Oh, there's one more question. Yes.
Thank you so much. Sorry, it's a bit of a very basic question, and I really enjoy all your presentations, although they are phenomenally above my head. My question is probably directed to Bathsheba, and it's about in our rare genetic syndrome studies, and this is not just in your field but also in other fields, we tend to see studies with a wide age range, looking from sometimes preschoolers all the way up to elderly. And how do you view that in your work in terms of, do you see that as a significant limitation? Do we know enough about the presentations across and at the trajectory of presentations, what we expect for a child versus a young person versus an adult versus a senior? And what are your thoughts on that?
Yeah, I'm going to repeat for Lynn real quick. So she asked about the age ranges in the Angelman study, and actually you can repeat it later. Oh, better.
You'll do that, yeah. So that's always definitely a consideration for the study. What we tend to see is the age of enrollment is usually between 4 and 12 years old. That's when participants usually come in for the first visit. And it's been a while to kind of try to get more adults to come down. The symptoms are very difficult to manage. It's hard to participate in a study like this for the older adults, but we are starting to collect that longitudinal data, and that really is the next focus of the study, to follow up with these participants and look at the different age groups.
Sorry, so you're looking at the different age groups. You look forward to sort of seeing how the profile changes as age
Exactly.
changes.
Mm-hmm.
Amazing.
Yeah, we try to do both types of analyses as the data come in. So both cross-sectional for ages and then also longitudinal.
One of the things that's happening with the natural history study, and Bathsheba has done just a phenomenal job of helping make all this happen, is that instead of requiring patients to come to the sites now, a lot more of the assessments are done remotely. Part of that we were forced into with COVID, but the model has also changed now so that where there are Angelman Syndrome Foundation supported clinics, patients can co-enroll when they're being seen for their clinical visit. If they're part of the natural history study, we can gather information. So it takes a little bit of the burden off, not having to come to Boston or San Diego, is that you can be seen in your local clinic. So we're trying to do things to make it easier for people to participate. And one of the reasons why enrollment, the numbers of patients that we've added has been lower is that we are trying to get more follow-up visits done so that we get that longitudinal data, which I think will be really critical for understanding many of these issues.
Well, thank you. I think we're over time and people are starting to trickle in, but thank you, Lynn, and